I and my Pfizer colleagues will now be happy to take questions.
Thank you, Dr. Gruber. Dr. Pergam is next, followed by Dr. Gans. Dr. Pergam.
Thanks, Dr. Gruber, for that presentation. I was curious, we haven't seen much of the data related to the dose finding in the initial phase 1 study. I'm curious if you can describe a little bit about whether there was a difference in immunogenicity between the 3 microgram versus 10 microgram. Is there a dose dependency in that particular comparison? I'm just curious based on differences in dosing of the vaccine.
Thanks for that question. Obviously, as I said, we paid strict attention to finding just the right dose that gave us the appropriate immune response after 3 doses. There are really 2 lines of evidence that support the basis for the dosing decision. One is the reactogenicity that I already shared with you. But the second one is really, if we can bring up the slide with the pink bars that speaks to the Omicron response. You may recall that while that's coming up, that we made decisions at the time of phase 1 based on comparisons to an adult population that gave us reassurance that for the reference strain we were likely to meet non-inferiority, right? Because that's the criteria for licensure. But in addition to that, if we can put slide 3 up, this is really the most compelling information, that having chose the 3 microgram dose and giving it as 3 successive doses, we now essentially nearly equal the sort of response that one sees in adults for which we know we have good protection against Omicron. So I think that confirms for us with the reactogenicity profile that I shared you and this finding that this is the right dose to provide protection. And again, although it's it's early, the 80% efficacy that we're seeing essentially matches what you would expect based on this type of response.
Okay. Again, Mike, just as a piece of clarification, did you see a difference in the 3 microgram versus 10 microgram in terms of antibody responses? It's just a clarification question.
The nature of the antibody responses were both above the responses that we saw in adults, but combined with the reactogenicity profile and knowing that for the reference strain we would be in a position to potentially meet non-inferiority, we chose that strain because we chose that dose because we again wanted to be confident that the vaccine would be accepted. Obviously, we already know that in older individuals, 30% of children are not getting the vaccine. There could be a lot of reasons for that, but one of them is the reactogenicity. So combined with that, and again, the data that I'm showing you here, it seems pretty clear that the 3 microgram dose is the right dose. And followed by, obviously, the phase 3 data that we have on reactions, which are very comparable to what we see in placebo recipients.
Okay, thanks.
Thank you. Moving on, Dr. Gans, followed by Dr. Portnoy.
Thank you for that presentation. I had a follow-up question maybe to Steve's question regarding the dosing, and I was looking at the slightly lower immune responses in the 2 to 5-year-old to the current dosing, and I'm just wondering, are there additional studies that actually may be looking at differing dosing and splitting these groups? As we know, it's a very large immunologic group, and there may be some nuances to how the toddlers or the preschool group is different than the infant group. So that's one question. I would also like you to talk a little bit about your breakthrough disease and see if there was any differences in the severity of disease in those who had received vaccine versus those who were in the placebo group. I realize hospitalization wasn't something, so that's not really what I'm asking. I'm asking about the disease profiles in those 2 groups.
Let me take your first question first about the nature of splitting the groups. I think the approach that we've taken is very consistent with every other age group in terms of dealing with the pediatric population. So for 12 to 17-year-olds, they get a 30 microgram dose. For the 5 to 11-year-olds, they get 10. And for now the 6-month to 5-year-olds, they receive 3 micrograms. And again, based on what we saw in the older children, where we had 19% severe fevers in the phase 1 group, we really don't feel comfortable expanding on that dose, and we don't need to, because we now see with the 3-dose series, which I think most people now agree, and we heard from Paul Offit yesterday, the importance of a 3rd dose in an Omicron era, that this is the right dose to immunize children to protect against Omicron. For the second piece, the nature of severity of the illness we've actually included in your briefing document, and there was really no difference if we looked at all the cases. So I'm talking about based on the number of symptoms that they had in terms of breakthrough compared to placebo, whether it was 1, 2, 3 or more symptoms. There really was not a dime's worth of difference between the respective groups, between those that break through that had received vaccine versus placebo. So that gave us confidence in that larger sort of population of cases that we have that does not appear to be an issue.
It also doesn't seem to, if the disease is the same in the placebo group, it didn't show a necessary advantage of the vaccine, I guess was what I was
I guess
asking.
the advantage of the vaccine is you're preventing the infection in the first place, right?
I get
so,
that.
That's the big advantage, right? But the good news is it doesn't seem to make the disease worse if, in fact, you've received the vaccine. And I think that's an important thing and why we looked at it.
Thank you. Dr. Portnoy, followed by Dr. Cohen.
Thank you. I guess I'm a little bit confused about this dosing in terms of micrograms, because your dosing is 3 micrograms, the Moderna dosing is 25 micrograms. Clearly, we're thinking in terms of micrograms the way we would think of proteins as a way of inducing an immune response, and yet the purpose of mRNA is to induce protein production. So is your mRNA just more efficient at making cells produce protein, or how should we think of micrograms in terms of the amount of spike protein that's produced by the cells? Can you kind of clarify that?
Yeah, I'll leave to Moderna to describe the nature of how they address their vaccine dosage. But I think, obviously, we don't have a complete understanding of the nature of the way that the vaccine works in terms of producing an immune response. So you have to go by the results. And the results are that in the setting of giving a 3 microgram dose, we had low reactogenicity compared to placebo, and after a 3rd dose, just as in adults at higher doses, we're getting an immune response that's comparable. It may well be that children, we've seen certainly that we are able to go down to a lower dose in children, and the expectation is perhaps they have a more robust response. That seems to be the case based on giving a 10 microgram dose to 5 to 11s and 3 micrograms to younger.
But have you ever measured the amount of protein that's produced as a result of the mRNA and how many cells are producing it and how persistent that production is for a given microgram of mRNA?
That's a pretty broad question.
Yeah.
I think that's obviously an interesting question to better understand the mechanism, and I would say it's somewhat academic in the setting of what we're trying to achieve here in terms of getting an immune response and a safety profile that's satisfactory, but worthwhile for people to pursue.
Okay, thank you.
Let me just, Dr. Jansen, the head of vaccine research and development, would like to make a comment about that last question.
Very brief.
Thank you, Bill. I think one important consideration for the answer to the question that was just posed is that the 2 mRNA vaccines are not created equal. They're actually very different vaccines. They use the same platform. They have different formulations. And so I think that's important to recognize. And the second piece is that we, of course, have optimized the vaccine for optimal expression of the antigens in cells. And if you ask the question, is there a large number of protein molecules expressed in the cells, the answer is yes. Thank you.
Thank you both. Dr. Cohen, final question.
Thank you. So I have this is maybe a multi-part question, but I'm wondering how you can be so sure of both your immunogenicity and VE estimates in the setting of the requested interval that you've requested between 2 and 3 doses is actually different than any of the is actually very different than the median in which your recipients received that third dose. So how can you be sure that they would have a similar response 8 weeks after the first dose? And that sort of goes back to, this time period between the second dose, where there appears to be very little to no effectiveness, and the third dose would essentially mean that these kids would not be protected at all for an additional 8 weeks. So I'm trying to sort through both, you looked at effectiveness in kids who were vaccinated at a longer period after that second dose, and if you have any kids or any data on immunogenicity after 8 weeks.
So thanks for the question. I think where we take a great deal of comfort is sort of comparing like to like. So the nature of comparing this 11 to 13-week comparison in the older adult population to the immune response we're seeing in the younger sort of translates to the likelihood that you're going to get similar efficacy with that interval. Likewise, I think it's reasonable to assume that if the interval were lower, you would compare to what we saw in adults as well. I think there were a fair number of individuals that did actually get the vaccine in the 8 to 13-week period that are obviously being monitored as part of our ongoing assessment of efficacy, and perhaps we'll learn something from that, particularly as time goes on and we have longer follow-up.
So, but to clarify, we don't actually do we have data on adults that are immunocompetent that were vaccinated 8 weeks after their second dose?
We, in terms of data, in terms of 8 weeks, I'm not sure that we actually have data that we've analyzed at 8 weeks.
Right. So essentially what you're saying, and I hear your confidence, which is great, but what you're asking for, this 8-week interval, is not the same and can't be compared to adults or to many of the kids who are in your study.
I guess, what has to, and Dr. Cohen, I appreciate the question. I think what has to be balanced here is a reasonable expectation that we'll have some level of efficacy, certainly seeing that we have 80% at 11 to 13 weeks, and the need to get children vaccinated as quickly as possible to essentially achieve that efficacy. That was part of the reason, as you know, that for our primary series, we looked at 21 days. We wanted to narrow that interval what was reasonably possible to have enough maturation of immune response that you get a good response after the second dose. If we lengthen out now the period of time after the third dose, and we're all hearing, and the data supports that you need a third dose for Omicron, you're essentially trading off a theoretical issue that maybe I'm not going to get quite as good efficacy for the notion of a child, having that period of exposure before they can get that third dose. So I think that is the sort of thing that has to be weighed.
Thanks.
Okay, thank you, Dr. Gruber.